To begin, let's ask ourselves this question. What makes your biological characteristics different than those of your friends, parents, or siblings? The answer is that your DNA is unique. DNA is the primary genetic material contained within your cells and in nearly all organisms. It's used to create proteins during protein synthesis, which is a multi-step process that takes the coded message of DNA and converts it into a usable protein molecule. While that may sound confusing, the process can be broken down into simple steps. The first of these steps is the one that utilizes DNA, and it's called transcription, which is the process of using DNA to create messenger RNA, also simply called mRNA. This mRNA is a molecule that carries DNA's coded instructions for making a protein. Let's break down the process of transcription and further explore DNA's role in protein synthesis. To understand the role of DNA in protein synthesis, we first need to understand the basic structure of DNA. DNA is constructed as a double helix. To picture this, think of a twisted ladder, as you can see in this image. At the front of this image, the molecule is unwound, so you can see the bonding pattern of DNA's bases, which are the subunits of DNA it is built from. There are 4 bases: adenine, thymine, cytosine, and guanine, and each is usually represented by the first letter in their name, A, T, C, and G, respectively. When combined, these bases form your genetic code. They are arranged on 2 different strands, or the 2 sides of the twisted ladder. The code on one strand dictates the code on the opposite strand because the bases pair up in a specific pattern. Adenine always pairs with thymine, and cytosine always pairs with guanine. This pattern is important because it will serve as the template for making our mRNA molecule. But first, let's consider this question. Why must cells produce mRNA? So far we've learned that DNA is the genetic material within cells, that DNA is used to build protein, and that DNA is built like a twisted ladder. However, we haven't talked about where DNA exists within the cell, and this is important. DNA is housed inside the nuclei of our cells. The nucleus is the central compartment of a cell, and it directs cellular activity. DNA cannot leave the nucleus. This is a problem because protein synthesis occurs outside the nucleus. So, how does the cell transfer the coded instructions from DNA to the area outside the nucleus where proteins are made? The answer is that it creates mRNA. This mRNA is smaller than DNA, and it can exit the nucleus. To build mRNA, cells use the sequence of bases within DNA. Now let's discuss exactly how this works. Suppose one side of our DNA molecule contains the following sequence of bases: TACTG-CCTAG. If we follow the pairing pattern of DNA bases, A with T and C with G, we can build the following mRNA molecule: AUGACGGAUC. Notice how in mRNA, cytosine and guanine still pair, but uracil, which is represented with a U, replaces thymine in bonding with adenine. As we touched on earlier, the process of making this mRNA from your DNA template is called transcription. Acting as a template for transcription is the role DNA plays in protein synthesis. The newly synthesized mRNA will leave the nucleus and be converted into a protein during a process called translation. You are unique because of your DNA, or the genetic material inside your cells. It's used during the process of protein synthesis, which is the multi-step process cells use to manufacture protein molecules. The first step of this process is called transcription, and it uses the base sequence of DNA to produce messenger RNA. Bases are the subunits of the DNA molecule. Messenger RNA, or mRNA for short, is the molecule that carries DNA's coded message outside the nucleus to be read and converted into a protein.